Hello everyone, I am Qazi Fariduddin. Again extend a very warm welcome to all of you for the plant physiology and metabolism course. In this lecture we are going to study the important aspect of photosynthesis, that is the difference between light and dark reaction, how Rubisco behaves in dual role. Now we will discuss what is photorespiration and then pathway of photorespiration or glycolate pathway. Now what is the significance of glycolate metabolism? Then difference between photorespiration and dark respiration. So all these aspects today we are going to learn. Now in my previous lectures we have studied in detail the light reaction and Kelvin cycle. So we might have gone through certain differentiating points between light reaction and Kelvin cycle. You see that the location of these two reactions, that is light reaction and dark Kelvin cycle, differ from each other. One occurs in thylakoids of chloroplast, whereas Kelvin cycle occurs in stroma region of the chloroplast. As you see that light reaction can occur only in the presence of light, whereas Kelvin cycle occurs both in the presence or absence of light. So you see that in light reaction, light energy is absorbed by photosystem 2 and first to start the reaction. But what happens in Kelvin cycle, that carbon dioxide is absorbed by the stroma to start the reaction. And in light reaction generally ATP, as you might be aware, ATP and NADPH are the end products, whereas in Kelvin cycle you see that glucose is the end product and ATP and NADPH are basically utilized, synthesized in the light reaction. And from the type of reaction, light reaction is basically photochemical phase, whereas Kelvin cycle is biochemical phase. So you see that how this light reaction and Kelvin cycle differ from each other, and once we know the striking feature of these two types of reaction, that is light reaction and Kelvin cycle, we should keep in mind these differentiating points in mind. And now the next aspect is Rubisco. Just I mentioned that how Rubisco plays dual activity. So just what do you mean by dual activity? So this Rubisco can perform dual function both in the form of carboxylation reaction, that is addition of carbon dioxide to 5 carbon compounds, namely ribulose bisphosphate, leading to the formation of a 6 carbon compound that subsequently break down into 2 molecules of 3-phosphoglyceric acid, and another is oxygenation reaction, that is addition of an oxygen molecule resulting in the formation of 1 phosphoglyceric acid molecule and 2 carbon compound, namely glycolic acid phosphate molecule. Hence this Rubisco enzyme is commonly referred to as RUBP carboxylase or oxygenase, or could be abbreviated as Rubisco, ribulose bisphosphate carboxylase or oxygenase. So you see in the slide that this dual role of Rubisco was very interesting, and in the year 1920 Otto Warburg, a German biochemist, discovered in chlorella chlorella is a green algae, that photosynthesis was inhibited by oxygen. This was the observation of Warburg. So, despite the photosynthetic origin of oxygen, it has been observed that a rise in oxygen concentration can decrease the rate of photosynthesis. So, this inhibition of photosynthesis by oxygen is known as Warburg's effect, and it was later recognized as light dependent release of carbon dioxide due to oxygenase activity of Rubisco. Basically, carbon dioxide acceptor known as RUBP, which occupies the active site of the Rubisco enzyme. Subsequently, either carbon dioxide or oxygen can attach to the enzyme, leading to either carboxylation or oxygenation process. As a result, you see there is a competition between oxygen and carbon dioxide for active center of the enzyme. However, it should be noted that Rubisco basically exhibits a much stronger affinity for binding carbon dioxide compared to oxygen. Therefore, in the presence of natural atmospheric concentration of these gases, where oxygen and carbon dioxide both are present in a different ratio, like oxygen is present in 21%, whereas carbon dioxide in 0.3%. So, what happens that the carbon dioxide, the atmospheric concentration of oxygen and carbon dioxide has very important, and you see that in this context carbon dioxide has significant advantages. So, the proportion of oxygenation in leaf photosynthetic gas exchange under these atmospheric condition is only 25 to 30%. However, when the carbon dioxide concentration decreases and the concentration of oxygen increases, then competitiveness of oxygen increases and Rubisco performs oxygenase activity, which will be discussed that the Rubisco, when Rubisco performs oxygenase activity will be discussed in photorespiration or glycolate pathway. Now, you see that question arises, what is photorespiration? So, you see that photorespiration is also known as C2 cycle or glycolate pathway, is a sort of metabolic pathway in plants that occurs in the chloroplast, peroxisome and mitochondria of the plant cell. And this process is basically initiated when oxygen competes with carbon dioxide for binding to Rubisco, leading to oxygenation of RUBP instead of carboxylation. This leads to the formation of one molecule of 3-phosphoglycerate and one molecule of 2-phosphoglycolate. However, what happens, 2-phosphoglycolate cannot be utilized in Calvin cycle. Instead, this 2-phosphoglycolate enters the glycolate pathway, also known as the C2 cycle, which, as I mentioned, which involves the chloroplast, peroxisomes and mitochondria. And the interesting feature of this pathway is the release of carbon dioxide from 2-phosphoglycolate, ultimately allowing plants to recover some of the carbon lost during photorespiration. Now, we will discuss in detail how this pathway proceeds. So, it is very important to note that the enzyme Rubisco can use either CO2 or O2 as a substrate. Rubisco basically adds whichever molecule it binds to a 5-carbon compound called ribulose 1,5 bisphosphate. The reaction that uses carbon dioxide is the first step of the Kelvin cycle and leads to production of sugar. The reaction that uses oxygen is the first step of photorespiration pathway, which wastes energy and undoes the work of the Kelvin cycle. It is very interesting. So, what determines how frequently each substrate gets chosen? Basically, two key factors are the relative concentrations of oxygen and carbon dioxide and temperature. These are the two key factors. When a plant has its stomata or leaf pore open, carbon dioxide generally diffuses in and oxygen and water vapor diffuses out and photorespiration is minimized. However, when a plant closes its stomata for just reducing loss of water by evaporation, oxygen from photosynthesis generally builds up inside the leaf. Under these conditions, photorespiration increases due to higher ratio of oxygen to carbon dioxide. In addition to this, Rubisco has also higher affinity for oxygen when temperature increases. At mild temperature, generally the tendency of Rubisco is to bind carbon dioxide about 80 times higher than its affinity for oxygen. Now, how the process of photorespiration proceeds? It begins in the chloroplast when Rubisco attaches oxygen to RUBP in its oxygenase reaction. Then the first two molecules are produced, a 3-carbon compound 3-phosphoglyceric acid and a 2-carbon compound phosphoglycolate. Followed to this, this 3-phosphoglyceric acid is a normal intermediate of Kelvin cycle, you might be aware, but phosphoglycolate cannot enter the Kelvin cycle. So, phosphoglycolate, which is a 2-carbon compound, is dephosphorylated by phosphatase and converted to glycolate inside the chloroplast. Glycolate then travels to the peroxisome, the next cell organelle, where it is oxidized to glyoxylate, which is also a 2-carbon compound, by enzyme glycolic acid oxidase. In basically peroxisome, hydrogen peroxide is produced, which is removed by activity of enzyme catalase. Now, glyoxylate is converted into glycine, which is a 2-carbon compound, with the help of enzyme glutamate glyoxylate transaminase. So, this is basically a transamination reaction. Then you see that glycine travels from peroxisome to mitochondria. So, in mitochondria, 2 glycine molecules from 2 reactions of the pathway react to form one serine molecule, which is a 3-carbon compound, and remaining 1 carbon is released as carbon dioxide molecule. Here also ammonia is released. Enzymes involved during this reaction is serine hydroxyl methyl transferase. Now, what is the fate of serine? Serine returns back to the peroxisome, where it is converted to hydroxypyruvate, which is a 3-carbon compound. This is basically a transamination reaction and catalyzed by the enzyme serine glyoxylate aminotransferase. So, in peroxisome, hydroxypyruvate is now reduced to form glyceric acid, and the enzyme involved is hydroxypyruvate reductase. Now, this glyceric acid or glycerate enters the chloroplast and is phosphorylated by glycerate kinase to form 3-phosphoglyceric acid, which is also a 3-carbon compound as I mentioned earlier. This 3-phosphoglyceric acid, as we know that it is a very important intermediate of Calvin cycle, thus enters the C3 cycle. So, photorespiration is basically a metabolic pathway occurring in plants, releasing carbon dioxide, decreasing photosynthetic output and limiting biomass production. In contrast, photosynthesis, which fixes carbon dioxide, photorespiration releases fixed carbon dioxide as carbon dioxide, utilizing ATP but not contributing to carbon fixation or generation of ATP production. As a result, this process is considered a wasteful process. The final product of photorespiration includes carbon dioxide, ammonia and a reduced carbon compound. In addition to this pathway, as you know, that consumes energy without contributing to essential processes. Now, what are the factors that affect photorespiration? Of course, environmental factors such as temperature and oxygen concentration influence photorespiration, which tends to increase under high temperature and low carbon dioxide. So, the bottom line is that hot and dry conditions tend to cause more photorespiration unless plants have special features such as in C4 and CAM plants to minimize the problem, which we will discuss in our coming lectures. In addition to this, some inhibitors of glycolic acid oxidase like alpha hydroxy sulfonates hinders, or you can say inhibits, the process of photorespiration. Now, question arises that what is the significance of glycolate metabolism? So, let us discuss in what ways photorespiration is a wasteful process. We have to justify it. So, you see that 1 in 10 carbon atoms from the carbon acceptor molecule, that is RUBP, is lost during the process of photorespiration. In addition to this, energy in the form of NADPH and ATP is also consumed during the assimilation of ammonia, adding to the energy cost of carbon dioxide assimilation. Photorespiration also increases the energy cost of carbon dioxide assimilation, requiring more photons. Generally, number of required photon increases from 8 to 14 for the process. The efficiency of carbon dioxide fixation is generally measured as quantum efficiency, which decreases due to photorespiration. Higher temperature generally further decreases the quantum efficiency because photorespiration increases more than photosynthesis, because the relative rates of carboxylase and oxygenase activity basically depends upon the affinity of Rubisco for carbon dioxide and oxygen, the ratio of carbon dioxide and oxygen present in the cell solution. Therefore, with an increase in the temperature, the solubility of carbon dioxide decreases more compared to oxygen, and the enzyme affinity for carbon dioxide decreases compared to oxygen. As a result, glycolate is produced more as compared to 3-phosphoglyceric acid, especially in C3 plants. So, this indicates that photorespiration is clearly a wasteful process. However, the glycolate produced in this cycle has some significance, like one is scavenging role of photorespiration you see in this slide. So, one of the significant role of photorespiration is believed to be its scavenging function, because glycolate production cannot be avoided due to inherent oxygenase activity of Rubisco during the photorespiratory cycle and evolved to remove glycolate with minimum carbon loss. About you see that 75% of the carbon loss from the Kelvin cycle as 2-phosphoglycolate, which is recovered in the oxidative photosynthetic carbon cycle. Out of the 10 carbons of RUBP, only 1 carbon is lost as carbon dioxide, while 9 are recycled as 3-phosphoglyceric acid. Then the next function is involvement of 3 organelles for glycolate removal. As you know that 3 organelles are involved in the removal of glycolate due to its toxicity to Rubisco and Kelvin cycle enzymes. Glycolate is transported out of the chloroplast and further oxidized to produce glyoxylate in peroxisome, not in chloroplast, so that to avoid inhibition of photosynthesis. Peroxisome efficiently removes hydrogen peroxide and convert glyoxylate to glycine. Moreover, substrate channelizing in a multi-enzyme complex prevents the release of glyoxylate, hydrogen peroxide and hydroxypyruvates, the intermediates of glycolate metabolism. Even if the peroxisomal membrane is damaged, the enzyme complex remains intact, indicating the protective function. Enzymes present in the cytosol, because they are present in the enzymes in the cytosol are present to convert glyoxylate to glycolate in case of any sort of leakage. Then third is role in protection from photoinhibition is also very crucial. What happens during high light intensity and temperature, stomata close to prevent water loss, which leads to decrease in carbon dioxide concentration as compared to oxygen. So, what happens? The decrease in carbon dioxide availability causes the accumulation of reduced intermediates of light reaction. Without carbon dioxide, ATP is not consumed, so the proton gradient across thylakoid is generally reduced, hence preventing the dissipation of photosynthetic electron transport. This leads to production of reactive oxygen species and damage to the photosynthetic apparatus, a phenomena generally termed as photoinhibition. Now, if these ATP and NADPH are consumed in some way, this damage to the photosynthetic apparatus will be prevented. So, basically plants employ photorespiration as a mechanism to consume NADPH and ATP, hence protect the photosynthetic apparatus from light induced damage. Now, how photorespiration plays a role in nitrogen economy? Photorespiration basically plays a significant role in maintaining nitrogen economy by conserving nitrogen. You see that when glycine is converted to serine in mitochondria, one nitrogen atom lost as ammonia is reassimilated in chloroplast through GS-Gogat reaction, thereby conserving the nitrogen. During ammonia assimilation, reducing equivalents are used as reduced ferrodoxin and NADPH. Now, role in amino acid synthesis, role of photorespiration in amino acid synthesis. The photorespiratory cycle is essential for the synthesis of two amino acids, namely glycine and serine. Alternate pathways for amino acid synthesis function when photorespiration is suppressed. Now, we will basically discuss the difference between photorespiration and dark respiration or usual respiration. But the detailed mechanism of usual respiration will be discussed in my upcoming lectures. So, how this photorespiration differs from usual respiration? You see that photorespiration is light dependent, whereas usual respiration is light independent. And in addition to this, this exclusively takes place, photorespiration exclusively takes place in green tissues of C3 plants and minimum in C4 plants, whereas the usual respiration takes place in living tissues of all aerobic organisms. Photorespiration basically linked to and entirely rely on the Kelvin cycle, whereas usual respiration is not inherently linked to C3 cycle, but you see that usual respiration and C3 cycle are spatially and temporally distinct process. They do not rely on each other. You see that photorespiration generally takes place in various cellular compartments, including cytoplasm, chloroplast, peroxisomes and mitochondria, involving glycolate pathway. Whereas usual respiration occurs exclusively in the cytoplasm and mitochondria, involving glycolysis, TCA and terminal oxidation. You see that photorespiration basically consumes oxygen and at 3 stages of release carbon dioxide, and at 3 stages, photorespiration consumes oxygen at 3 stages and releases carbon dioxide only at 1 stages. So, but you see, as usual respiration, oxygen is solely utilized during terminal oxidation, whereas carbon dioxide is liberated at multiple points. So, you see that in other aspects, photorespiration and dark respiration also differ from each other. Generally, for every molecule of carbon dioxide released, one molecule of ammonia is liberated during photorespiration, but in usual respiration ammonia is not generated. So, you see that these two processes, that is photorespiration and usual respiration, significantly differ from each other. So, in this lecture we have basically learned the difference between light and dark reactions, the dual role of Rubisco as carboxylase and oxygenase activity, the consequences of oxygenase activity of Rubisco in photorespiration, leading to the wasteful consumption of ATP and release of carbon dioxide. Further, the significance of glycolate metabolism in conserving about 75% of the carbon lost in Kelvin cycle has also been well explained. So, this is all about for today's session. Thank you very much.